Hello guys, welcome back to Hot News Studio and welcome back to our segment Science and Technology. Today we are going to talk about the Nobel Prize winner in Physiology or Medicine. In a remarkable turn of events, the Nobel Prize in Physiology or Medicine for this year has been awarded to the groundbreaking messenger RNA vaccine against COVID-19, which have not only transformed the course of the pandemic but also saved countless lives. This prestigious honor goes to pioneering work of Kathleen Carrico and Drew Weissman, who have significantly advanced vaccine development and deepened our understanding of how messenger RNA interact with our body's immune system. When Drew Weissman received the news, his emotion took him on roller coaster ride. He described, "I am going through a series of steps. It started off with an incredible joy and surprise, and right now I am pretty much numb." The journey of Carrico and Weissman began in the 1990s when they joined forces at the University of Pennsylvania to study in vitro synthetic messenger RNA technology. Their groundbreaking 2005 paper showcased their ability to deliver modified messenger RNA into the body, prompting an immune response that educates the immune system to combat future viral infection. Over the years, their work on messenger RNA vaccine addressed the critical challenges, including the body's inflammation response involving the production of harmful cytokines. When the COVID-19 pandemic struck, their messenger RNA technology paved the way for highly effective vaccines against SARS-CoV-2, the virus responsible for COVID-19, and these vaccines were well suited for large-scale production. Gunilla Carlsson Hidemstam, a member of 2023 Nobel Committee for Physiology or Medicine, emphasized the significance of rapid vaccine development due to improvement in technology and foundational discovery. She stated, what important here is that vaccine could be developed so fast. Kathleen Carrico, born in 1955 in Hungary, has had a remarkable career. She served as a senior vice president at BioNTech RNA Pharmaceuticals, a major manufacturer of mRNA COVID-19 vaccine. I currently act as external consultant for BioNTech. She is also a professor at the University of Szeged in Hungary and an adjunct professor at the Perelman School of Medicine at the University of Pennsylvania. Drew Weissman, born in 1951 in Lexington, Massachusetts, in the Robert Family, professor in vaccine research at the University of Pennsylvania and the director of Penn Institute for RNA Innovation. Kathy Newsel, a professor of vaccinology and the director of the Center for Vaccine Development and Global Health at the University of Maryland School of Medicine, noted, "The award is a victory for vaccine and their potential to advance health and promote equity." Historically, vaccines were often created using weakened or inactivated whole viruses. However, in recent decades, the researchers have explored smaller viral components like viral genetic materials such as DNA or RNA. When Carrico and Weissman introduced foreign in vitro mRNA into human cells, they discovered that it could trigger a strong immune response, leading to production of protective antibodies. Although the scientific roadblocks, skepticism, and funding challenges arose, they persevered in their quest for solutions. Weissman reflected on the difficulties they faced. It was a constant battle for 25 years. He struggled to secure funding. The Carrico faced setback. Conducting this research was extremely challenging. But we recognized the potential early on and understood the importance of RNA. The determination kept us going. We never gave up. The team overcame these obstacles by finding ways to modify mRNA to be less inflammatory, such as replacing uridine with a similar molecule called pseudo-uridine. They also developed a more efficient delivery system that employed lipid nanoparticles to protect mRNA and aid its entry into the cell for protein production. In the early 2000s, Carrico and Weissman conducted animal trials with mRNA vaccine for various pathogens, including Zika, influenza, and HIV. Weissman noted, "In nearly every animal model we examined, we achieved 100% protection." This research opened a new path for therapy and vaccine development, which proved to be crucial during COVID-19 pandemic. As SARS-CoV-2 virus began to spread globally, Weissman and Carrico mRNA research quickly became the basis for COVID-19 vaccines. The mRNA vaccine approach offered several advantages, including the need for only the pathogen sequence without the necessity of growing and inactivating a virus. Wiesmann explained it's a straightforward procedure because it's an enzymatic reaction. It took only 2 months from the release of the sequence to the first patient receiving the vaccine. Clinical trials, vaccine production and distribution expanded rapidly, with the companies manufacturing hundreds of millions of doses within a year. Carrico highlighted transitioning to COVID was a straightforward process, it was already prepared. The mRNA COVID-19 vaccine worked by injecting genetic material specific to SARS-CoV-2 spike proteins, the surface protein that allow the virus to attach to healthy cells. Cells decode the modified mRNA in the vaccine and produce these spike proteins, enabling the immune system to recognize and neutralize the actual virus in the event of future infection. Kathleen Nutzel, who has also been involved in mRNA vaccine research for malaria, noted, "We are emerging from the worst pandemic in over a century, and these vaccines have significantly contributed to saving lives and reducing illness. I believe this technology, adaptability and flexibility have the potential to be transformative, especially for low and middle income countries." Looking ahead, mRNA technology offers a broad range of application. Kathleen Carrico's initial interest in mRNA research was focused on therapies rather than vaccines. She stated in 2021 interview, "I was making modification to RNA because I always wanted to develop it for therapeutic processes." While mRNA technology has played a pivotal role in addressing the COVID-19 pandemic, it holds promise for numerous other medical conditions. Nick Sanders, a principal investigator at Ghent University Laboratory of Gene Therapy in Belgium, emphasized that a vast number of individuals will benefit from this technology. It can be used to treat any disease caused by a malfunctioning protein, allowing patients to produce their own therapeutic protein. Sanders stated, "Nobel Prizes with such a profound impact on society are rare, occurring only once every 25 or 50 years. Wiesmann, Carrico and other research groups are actively exploring the application of this technology to autoimmune diseases, cancer, food and environmental allergies, bacterial diseases and insect-borne diseases." In a notable development, Wiesmann's team published a paper in Science in July, demonstrating the direct delivery of RNA gene editing machinery to bone marrow stem cells. This breakthrough holds promise for treating diseases like sickle cell anemia, where stem cells are transitionally extracted and cultured, treated and reintroduced into the body. Wiesmann anticipated that mRNA treatment will be available for sickle cell anemia patients in around a year and a half. His team is also conducting mRNA clinical trials, including a phase 1 trial for the disease amyloidosis and vaccine trials for HIV, norovirus and malaria. They are planning to launch clinical trials for a pan-coronavirus mRNA vaccine soon, with a potential to prevent future coronavirus epidemics. In the words of Drew Wiesmann, "The future is now. These therapeutics are already making a difference in people's lives." So that's it for today. Thanks for watching this video. If you like this video, please do like, share and subscribe our channel. We will meet again in the next video. Thank you.